Okay, perfect. So, hi everyone, welcome to the QLS seminar. It's our pleasure to have Alex Diaz-Papkovich, who is actually a QLS alumnus. So, Alex did his PhD in QLS with Simon Ravel, where he studied topological data analysis methods for genetic data. And now he is a postdoc at Brown University, where he is at the Data Science Institute working with Sohini Ramachandran. Hopefully, I got that right. And well, Alex, take it away.
All right. Thank you, Benjamin. Yes, you did get it all right. So today I'll be talking about low-dimensional representations of high-dimensional biobank data. So just to give an outline of this talk, split up into, I think, four main chunks. Basically, I'll talk a little bit about data in the biobank era. Then I'll talk about some of the research that I did through my PhD and in my postdoc, which focused on dimensionality reduction, clustering, and visualization of biobank data and its applications. Some new stuff that I've been working on, the ethical and social implications of all of this, and then some of the conclusions in the future work. So just to keep in the back of your mind during this presentation, there's this one question that I always have, which is how do we visualize genetic data? So this is one of my papers that I wrote in 2019, and this is the same genetic data from the Thousand Genomes Project, but it's presented in four different ways. In the top left, we have principal components analysis. In the top right, t-distributed stochastic neighbor embedding, TSNE for short. In the bottom left here, we have UMAP, which is run on the genotype data. And in the bottom right, we have UMAP, which is run on the top 15 principal components of the genotype data. So I'll go more in depth into this later, but just to kind of give you a motivating thought, it's that we have the exact same data here, but we have it represented in four different ways. So we're currently in the biobank era. There's a lot of promises to the biobank and all the biobank data that we have. DNA sequencing has gotten super cheap, super efficient. So we now have sequencing data of millions of individuals, tens of millions probably at this point. And there's a lot of things we could do with this. One of the big promises is personalized medicine. So, in theory, we could develop drugs or medical interventions that are tailored based on your DNA. We could also study the genetic architecture of complex traits. So this would really help to develop knowledge of the biological function of different parts of the human genome. How are lungs formed? How does disease happen? Sorts stuff like that. And we can also study population histories. So we can look at things like human evolution. We can look at natural selection, migration patterns, historical events. So, big waves, migrations, wars, things like that. Uh, but there's also sort of the perils of the biobank. So one of the problems, which is a lot of the work that I've focused on, is that this is very complex, high-dimensional data. So the human genome is massive to begin with, but you also have tons of environmental variables that are in there, lots of social and cultural factors that go into measuring human health, stuff like that. So there's a lot of confounding and interaction between genetics and the environment. It can be really difficult to parse subtle signals, so this is kind of a data mining problem, and this has been around in many fields for a while, but biobanks have it as well. And then in the context of population and statistical genetics, we often talk about these populations, but these populations are often not known in advance. A lot of biobanks, they just kind of they have very different sampling methodologies. The UK Biobank takes like 500,000 people who live in the UK. Some of them are volunteer based. Some of them are like formal simple random samples or stratified random samples. But usually we don't know what populations we're looking at in advance. So that can be a little challenging when you want to talk about the health of a population, for example. One of the other problems I focused on a lot is this idea of structure in genetic data. It's called population structure. So, if any of you have worked with Simon Gravel, if he's taught a module, or if someone else has taught a module in QLS on population genetics, you've probably taken a look at population structure. But it's basically the systematic differences in allele frequencies, and these can act as confounders in models using genetic data. But they can also be pretty informative because, those patterns can tell us about the history of a group. And then one of these other things that's coming up for me a lot is what are the social consequences of genetics research? Because we have so much genetic data, we have so much genetic research now, and it's available online for free, accessible to anyone a lot of the time. How is this impacting how people think about the connections between genetics and different groups? So I'm going to introduce some of these core concepts. So this is genetic data. Here's an example of what it can look like. So it's often this huge matrix of the number of non-reference alleles. So, we have two chromosomes. So the number of non-reference alleles is going to be 0, 1, or 2. As you can see, like this is a huge matrix. Like this is just an example of what it looks like, but it's very, very high dimensional a lot of the time. You might have something like the UK Biobank where you have 500,000 or so individuals, and then, you might have a genotype array of like 900,000 single nucleotide polymorphisms. So this does require dimensionality reduction if we want to do any sort of meaningful analysis on it, because it's just a ton of signal to parse. One of the frameworks that I work in is this idea of exploratory data analysis. So there's this great diagram in a textbook I really like called Modern Statistics for Modern Biology. Um, but essentially, it kind of posits these two different approaches to statistical analysis. And these two frameworks have been around since the 70s or so. Fisher's paradigm has been around since like 1900. But the idea is you have a pretty straightforward and linear approach to science where you have a biological question, you develop a hypothesis, you design an experiment around this null hypothesis, you collect the data, you compute a p-value, and then you reach a conclusion based off of that. Tukey, who was a statistician in the 60s and 70s, he proposed a different framework, which was exploratory data analysis, which is where you have the data already, and then you sort of develop hypotheses and analyses based around that. And in fact, this is a pretty common paradigm that we find ourselves in now with biobanks, because generally, if you're working with biobank data, you're not the one who collected the data. You didn't develop the sampling methodology or anything like that, but you have it and you want to do an analysis. But the idea is that you have your data, and then there's this whole chunk of the analysis that's dedicated to visualizing it. So you want to present your data in some way. You want to look at the graphics. You want to look at form recognition. So do you see clusters? Do you see clumps of data? Do you see unusual patterns? And one of two things can happen here. Either you can say, oh, this is just an artifact of the data, so I'll do some diagnostics on it. Or you can develop some hypotheses and say, I think that this is because there's some underlying factor here. And then once you develop those, you go into confirmatory analyses and you develop some conclusions. So topological data analysis, this is a statistical method that's motivated by qualitative geometry. So you work a lot with these ideas of shape, of connectivity, of nearness, of similarity. You work with point clouds and clusters a lot of the time. And if you want to think about it a little intuitively, you have this idea that perhaps these different data points are near each other in some sort of space. But it's not necessarily obvious because it's not just, it's not something in a 2D Cartesian plot. If you're working with 900,000 dimensional SNPs, you might intuitively have this idea of two samples being near each other in a genetic space, but it's not as obvious as it would have been before. But the idea is that topological data analysis can let us kind of use these ideas. And importantly for me, it lets me generate visualizations that allow me to study and interrogate the data. So it's closely connected to the idea of manifold learning, that there's an underlying manifold to the data that you work with. And one of the methods that I really use that I'll be talking about is UMAP, which is a nonlinear dimensionality reduction approach that is rooted in topological data analysis. And then I'll follow up with a clustering algorithm that's very closely related as well. So population structure, as I mentioned, it's the systematic difference in allele frequencies between groups. It's this idea that there's non-random variation between populations. So it's a confounding factor in genetic analyses, but it can be really informative of population histories. There's lots of methods that exist to identify it or adjust for it in models. So you have principal components analysis, linear mixed models, UMAP and clustering, which is the stuff that I've worked on through my PhD and my postdoc. And visualization is actually a really good way to study population structure. So talking about some of the motivations behind my research. So there's the first one, which is to develop a dimensionality reduction framework for biobank analyses. The reason I want to do this is to give researchers a better understanding of their own data. You have these huge complex data sets and you have a lot of questions like, is there a genetic architecture behind a lung phenotype or something like that? Are the distributions of blood cell densities different between different groups of people? That can be hard to answer in general, but a little easier with a dimensionality reduction framework. And then also just to understand the sources of the population structure within your data. If you sample like 20,000 Canadians at random, we have a pretty diverse immigration history, as well as like indigenous history here. So, if you want to look at, okay, what are the populations that we have within Canada, a dimensionality framework for a population like ours would be really good to sort of say, okay, well, we can study the histories and figure out the sources of population structure in the data. Another one I'm really interested in is studying the overlap between genetic and environmental data. And the idea is that I want to quantify the relationships between population structure and environment. Because often you'll find that, a classic example is an ethnic enclave within a city where the population structure is basically geographical. They all tended to come from one part of one part of Europe, and then they moved into Montreal and established a neighborhood there, and they've lived there for like 3 generations. But there's also obviously going to be a heavy overlap between genetics and the environment there. So the idea is that if we use visualization dimensionality reduction, we can study this overlap, maybe quantify it in some way, and take a look at what some of the factors are in population health, for example. And then the last one is to understand how genetics research exists in the information ecosystem. Because now, especially with the popularity of things like ancestry testing, um, genetics has really, it's always been kind of tied to this idea of group identity, like I am a Cuban and, I can trace my ancestry back this many generations. But now we can say something like, oh, I'm like 47% this and 53% something else, which, there's a whole body of research behind that now. And I've really been wondering a lot. In particular, how does this kind of stuff exist in the information ecosystem? So in the internet, how does the world in general understand the associations between genetics and ancestry and health and things like that? So for those who've seen me talk before, these first 2 points, it's going to be a little bit of a review because I gave a talk last year, and this third point, this is going to be all brand new stuff. So principal components analysis, PCA for short. This is a method of linear dimensionality reduction. For those of you who remember linear algebra, the idea is you take your data and you reduce it to the lowest number of, or you reduce it to a low rank dimensionality that maximizes the variance explained. So principal component 1 explains the most variance in the data. Principal component 2 explains the second most variance in the data, and so on in that manner. It's very, very common in population genetics. So on the left here, this is the 1,000 Genomes project dataset, and this is what principal components 1 and 2 look like. So, in this case, you have these populations tend to be African ancestry. These ones over here in the bottom left corner tend to be European ancestry. These up here are East Asian, and then you can see there's some green in the background, which is actually principal component 3 in the third dimension. Those are the South Asian populations, and then you have these pink dots. These are Central and South American populations. So you can see it kind of reflects patterns from geographical ancestry. So it reflects things like geography, things like admixture. These patterns really come up very nicely. The other thing that these coordinates are useful for is if you run a genome-wide association study, or GWAS, you can actually add them as covariates into the model, and they can help to adjust for population structure to make sure we don't have any spurious relationships between a phenotype we're measuring and the genetic work or the genetic data that we're using. So there's a very famous figure in population genetics by John Novembre, and the idea is that principal components also reflect what's called isolation by distance, which means that genetic differentiation between populations in a lot of contexts is roughly proportional to the distance. So in this case, what he did was he took the genetic data of a bunch of European individuals, carried out principal component analysis, and then overlaid them on a map of Europe and found that they actually correlated very strongly. So PCA is a fantastically useful tool in population genetics. It's got a lot of applications, and as you can see here, it makes really cool figures too, and it remains popular, and I still use it all the time. But one of the drawbacks is often if there's a lot of signal in the higher order PCs, so beyond principal component 1 and 2. So on the left here, this is from a CBC article where they plotted the top 3 principal components, and 3 dimensions don't work well in a two-dimensional, piece of paper or website or whatever. And the other thing is, on the right here, I have a figure from a paper that I wrote where we found signal of a cluster in principal component 7 or something like that, which required a bunch of pairwise plots. And I'm cheating a bit here because I actually used UMAP to find the signal and then traced it back to principal component 7. But the takeaway I want you to have from this slide is that the principal components, the signal actually often goes well beyond the top 2 PCs. So talking about UMAP, UMAP stands for Uniform Manifold Approximation Projection. The idea is that unlike PCA, which maximizes the variance explained in the low rank representation, it actually just preserves local neighborhoods and patches them together and presents the local structure in the data with some global context. So there's 2 parameters. Basically, you tell UMAP, I want to choose the number of neighbors that I want to preserve within a neighborhood, and I want to specify a minimum distance in a low dimensional space. And the more important parameter here is the neighborhood size. So do you want to preserve the nearest 15 neighbors of every point, or the nearest 5 neighbors, or the nearest 50, or the nearest 100? The idea is you would take these points and you would take them from the high dimensional space, build the neighborhoods, and then create a low dimensional representation that tries to be faithful to those neighborhoods, and you piece them all together. Um, so if you have a lower minimum distance, you get tighter and more dense clusters, which is not so great for visualization, but it's good for automated clustering, which I'll get into later in the talk. And the other benefit is we can reduce it to an arbitrary number of dimensions. Most people work in 2D, but UMAP can work in like 10 dimensions if you really feel like it. An HDB scan, I'm going to gloss over it a little bit, but basically it's a density-based clustering algorithm. And the idea that is that you can use it in general. I actually use it by pre-processing data with UMAP and then applying HDB scan. So I'll get into that later in the presentation, but just so you know that that's what's coming up. So a lot of the work I've done is on dimensionality reduction, clustering, and visualizing of biobank data. So within population genetics, UMAP focuses on the local structure in the data rather than the global structure. This makes it really, really good at finding clusters of data at different scales. So in particular, you can find the clusters of like continental level ancestry or even like city level ancestry sometimes. You can run UMAP on genotype data or the top principal components. And if you're interested in this kind of stuff, I have a paper from 2019. So it's got like 65 supplementary figures and a bunch of really good explanations in there. So that's a really good primer for it. And I'll be referencing it repeatedly. So like these figures are from that paper. But if you run UMAP on the 1,000 genomes data, on the left here, you can see that running it on the genotype data itself, you get roughly continental level ancestry. So you see this green blob here is sort of South Asian ancestry. This sort of orangish, yellowish, brownish one is more African ancestry. These groups here are European, the European populations and the Central South American ones. And then these purple dots, these are the East Asian populations. The other thing you can do is you can run it on the top principal components. So on the right here, I ran it on the top 15 principal components of the 1,000 genomes data, and it split it up into a bunch of very different fine scale clusters. So one thing to keep in mind is that the long distances between the clusters, these aren't really meaningful, but the actual clusterings themselves, those are what I find interesting. And also, you can see that pre-processing here does make a difference in terms of the final product. Now, if you run it on a huge biobank like the UK biobank, so 500,000 people, you get a figure that looks something like this. So, it's very rich in signal. So the idea is I took the top 10 principal components of the UK biobank. In this case, I chose to color them in by the selected ethnic background just to get some idea of like what is the composition of this biobank in terms of the populations. And you can see there's really complex patterns that are visible here. I won't go too in depth into them, but I mean, I think you can take my word for it that there are patterns and they're probably very interesting. But you see that there's really fine scale structure represented in here. So you can see like these clusters get really, really tiny. So there's little like gray blobs here that tend to reflect geographical ancestry, but they show them in the context of the whole biobank. And there's a lot of admixture in this biobank as well. So these individuals here, these huge blobs at the peripheries, these tend to be populations that were born in this huge one here born in the United Kingdom or Ireland. These ones here tended to be born in Africa or the Caribbean. These up here in India or Pakistan, and then here this is sort of East and Southeast Asia, and the individuals in between tend to have more admixed ancestries. So it's a really fantastic visual just in terms of being able to see all of the biobank at once and seeing a lot of the patterns at the same time. But I actually like to color these figures in by a whole bunch of different data that I have. So in this case, I actually had it's called northing and easting, but it's basically how far north or south you live in the UK and how far east or west. So you can see that the population structure also really reflects where you live in the United Kingdom. So all of these sort of bands of colors and clumps of colors, they tend to represent more geographically concentrated individuals. You can color in by phenotype data. So on the left here, I have height. On the right, I have a leukocyte count. So that's a white blood cell count. And you can see that there are differences in the clusters in their colorings because on average, some groups tend to be taller or shorter, or they tend to have higher or lower blood cell counts. So this is all very relevant and interesting if you're studying, say, biomedical traits. You might want to develop tests for what is a normal blood cell count for an individual, or perhaps if you're studying, just what is the distribution of some variable. It's really fantastic because it gives you so much context. So UMAP became very popular in genomics, and I'm going to put an asterisk here because I'm going to return to this point. So after my paper was published, on the left here, this was Nomad. This is an American data set. They ran UMAP on their data, and then they published that figure because they found it was a useful explanation of the data. This middle one here is Biobank Japan, and as you can see, they split it into two clusters, which is, it's a little questionable, but I'll come back to it in the next slide. And then on the right here, this was the All of Us biobank, which also ran UMAP, and then shows an interesting coloring scheme that generated some discussion. So I will return to these last two, but just as a preview. So before I get to that, I want to talk about the clusters that we see in UMAP. So you can see here, this was a Biobank Japan paper. And the idea is they wanted to use these clusters in their polygenic scoring, and they wanted to test, is there a difference in polygenic score values based on running analysis on this mainland Japan cluster versus the non-mainland Japan cluster, so some of the smaller islands that were outside of Honshu. Um, the clustering method here is certainly informal because the idea is, you have this big cluster on the left and then you had a bunch of small ones on the right, and they literally just put a dotted line in between them and said, okay, we have cluster one, cluster two. And one of my questions was, okay, well, that's one way to do it. Can we do this better? Because you can obviously, there's banding. You can see that there's banding in this cluster here. These clusters have lots of subclusters. So I don't think that this is the ideal way to extract clusters from data. Um, so the thing is, UMAP, it generates clusters. And I spoke to Leland McInnes, who's the developer of UMAP, a couple of months ago. And he actually said that one of his motivations behind UMAP was to find clusters in data. But that being said, UMAP is not a clustering algorithm. It doesn't actually give you cluster labels. It doesn't extract them. You can see them, but you can't get them. So, the two questions I had were, can we extract these clusters algorithmically? And if so, what could we use these clusters for? So this is where HDB scan comes in, and this is a hierarchical density-based clustering. And the idea is for this algorithm, you just specify the minimum points in a cluster. And uh, sorry, just to take a step back, the way this algorithm works is it looks in a space and it finds dense areas of data. So if you can imagine there's a big space somewhere, and then you have 10 points that are really tightly clumped, it would consider those to be a cluster. So what you actually specify is the number of minimum points that you want in a cluster. And if you don't have enough points, the algorithm just discards them as noise. This is in contrast to a lot of methods like, say, k-means, where you say, I want 10 clusters and I want you to classify all the points in my data into these 10 clusters. So what I found really nice for HDB scan is when I'm working with a biobank like the UK Biobank, I don't know how many populations I want. There might be 12, there might be 78. I don't know which number to choose. But I like to take the exploratory approach of let's just look for areas in the genetic space that are really dense with points. So there's actually a version of HDB scan that works particularly well with biobanks, and this is the Epsilon hat update. So the idea is that you can actually find really large, dense clusters of data and really small, less dense clusters. And this actually describes biobank data really, really, really well. Because the UK Biobank, which I've mentioned a few times now, has 500,000 individuals. I think something like 460,000 or 470,000 are born in the United Kingdom or Northern Ireland. So that is a huge proportion of the individuals that have population structure that is very similar and very local. At the same time, you have the rest of the individuals in the biobank who tend to be from all over the world. Because if you know British colonial history, the British Empire was massive. So, an ideal algorithm would be one that can look at data and say, okay, we might have a huge population that's super dense with data, and we might also have a bunch of smaller populations that are less dense. And just after having done all my PhD research, it turns out that this was a really good way to identify those different scales of data. The other nice thing is it's unsupervised and it's really fast. So the idea is if you run UMAP on your data and you run HDB scan after that, HDB scan runs in like 10 seconds on the UK biobank data. UMAP, depending on your processing power, will take like 1 to 20 minutes to run on the UK biobank principal components, but I mean, that's still super fast considering how huge the data set is. So this is kind of the paradigm that I cooked up. And the idea is you use UMAP in two ways. So first, you use it for visualization. So you can think of this as like a map. And you do this in two dimensions, and you optimize it for visualization. Um, and the second way that you use it is for clustering. So you run UMAP separately. Like you take that 2D map that you've created, and you just set it aside for a moment. Now, a map without labels is not very useful, so you need to generate these labels. So what you do to generate those labels is you run UMAP in higher dimensions. So if you'll recall, you can reduce data to an arbitrary number of dimensions with UMAP. So you could reduce it to like 5 dimensions or 10 dimensions. My recommendation is at least 3 dimensions, and then you optimize the parameters for clustering. So you make super tight, dense clusters. And then using those cluster labels, you can actually color the plots that you created in 2D. And this, this second point, I really want to highlight it because most people still only use methods like UMAP in 2 dimensions, which is great for humans because we can only perceive things in 2 dimensions or 3 dimensions. Computers don't have that liability. They can work in 5D, they can work in 10D. So, there's lots more dimensions to explore. So, let's take a step back and say, okay, I have my 1,000 genomes data, and let's pretend I lost all of the labels for it, and I run UMAP in 2 dimensions, and this is what I get. I can see there are clusters here, but I don't know what they are. So we can see these clusters, but how do we extract them? So the idea is we run UMAP again, put it in like 3, 5, 10 dimensions or something like that, and then we see what HDB scan extracts. And on the left, this is what we get when we color the 1,000 Genomes Project data by HDB scan labels, so the cluster labels, versus on the right where we color them in by 1,000 genomes labels. So, we found our population labels. Hurrah, we can compare and see what the concordance between these two is. And you can see visually like they're quite similar. And in fact, if you compare the clusters to each other, you see that the clusters have nearly 1-to-1 overlap a lot of the time. So in the 1,000 Genomes Project, there's good agreement between the cluster labels and the population labels. And this is kind of expected because the 1,000 genomes are basically 26 disparate populations from all over the world. So the population structure tends to really, really reflect the sampling structure. So when you run a method like HDB scan or UMAP and HDB scan on the 1,000 genomes, it's going to reflect that sampling methodology. Now, what was really fantastic was HDB scan ran on the UK biobank. So I did the same thing. I had my UK biobank figure, but I decided to color it in by the HDB scan clusters that I generated. So there's a preprint that I have here, which I also had a lot of fun writing. So I do encourage you to look at that one. But this one generated, I think, I forget the exact number, but probably like 26, 27 clusters in the UK biobank. It ran in a couple seconds. The one thing is no other method that I know of can really do this effectively. But the other bonus is that I don't need to identify these labels in advance. I can just generate the clusters using UMAP and HDB scan, and then I can study them. In terms of how to study them, it's kind of like the sky's the limit. You have all this sort of auxiliary data that you can use. Oh, sorry. I should add here that the cluster labels, so I mentioned that I extracted 26 clusters. What I said before still holds here. If you change the parameterizations, you're going to get different cluster labels. So you might end up with like 30 clusters or 40 clusters or something like that. But it does end up being a very useful way to study the data at different scales. But in terms of characterizing the structure, I have a lot of data in the UK biobank, so country of birth is one of them, and another one is the selected ethnic background. So what I did was something really simple. I just took a look at some of these clusters on the left here, and I decided to create word clouds based on the country of birth. And as you can see, there's a small cluster of individuals who were born in Somalia. There's another one that were born sort of in the greater Horn of Africa area, Eritrea, Ethiopia, Sudan. Some that are from Ghana, Nigeria, the Caribbean, and then a lot that are from Sub-Saharan Africa as well. And, we can also overlay this with a variable like selected ethnic background. And it's interesting because you can see that there's a sort of differential response where, in one cluster, you might have like 70% choose the select or they select the ethnic background African, and then the remaining 30% would say like, well, no, I'm other, I'm something else. And then you might have another cluster like here where there's a lot of admixture, so you can see that there's a whole variety of identities that people select. So it's interesting to show sort of the richness of the data and also to highlight that, yes, we do have a cluster, but there's obviously a lot of heterogeneity within that cluster. Um, if you'll recall for a moment, you can color in a UMAP by the phenotype distributions that you have, and you can see patterns and phenotypes. So in GWAS, what we do to adjust for this structure is we use the principal components. So if you'll recall what a linear model looks like in a genome-wide association study, we'll see that we have a phenotype, we have our genetic data, and the estimated effects, so that's the betas, and then we just add the principal components as a covariate in the model. But one question I've always had is what is the effect of these principal components? Like, what are we removing? How can we visualize this? So let's say we're looking at something like neutrophil count, which is a blood cell, and we remove the effect of the top 40 principal components. We find the average value of neutrophil count within the cluster, and this is an important part. So rather than just using one clustering set of parameters, I did this, I think, with 600 or so. I did a grid search and I smoothed over the entire data set. And then I decided to color it in by, okay, even after all this smoothing, even after removing the effects of the top 40 principal components, what is the distribution of neutrophil count in these populations? And you can see that, some populations here, they still have a higher than zero average for neutrophil count, and then some have sort of a lower one. But the key takeaway from this figure is that you've removed the top 40 principal components, but there's still evidence of population structure. Whether that's meaningful and what to do with it is a different question, but it's still there. And there's a lot of papers that would say take out the top 10 PCs, the top 20 PCs, the top 50 PCs, or something like that. But these are effectively arbitrary. So rather than saying, okay, we're going to remove the certain number of top PCs, why don't we just look at it and see what the effects are? So visualizing your data is extremely powerful. And this holds across every phenotype that I've looked at. So in here, this is FEV1, which is a lung phenotype. FVC, this is another lung phenotype related to FEV1. Height, you can see that these patterns, they still appear everywhere. And one of the things I decided to do was, well, what happened? Oops, that should say one PC at a time. Apologies to any Tims in the crowd. But what is the effect of removing one principal component at a time? So here we've started with the FEV, the forced expiratory volume phenotype, and this is with no principal components taken out. This is just how does the average of all of the clusters over all of the clusterings compare? And when you take out one PC at a time, you can actually see how the clusters sort of individually adjust, which I think is a really neat way of visualizing the effect of the top 40 principal components. Because you might have something like perhaps some signal doesn't appear or disappear till principal component 17. How are you going to find that? That's tough to figure out, especially across a lot of phenotypes. But this is kind of an interesting approach. One of the things we could potentially do is define populations for study. So the idea is polygenic scores. You model a phenotype and you predict it based off of the genotype. These scores are not, or these are known to not transfer well between populations, especially very differentiated populations. Uh, but one of the questions I had is this true for all phenotypes or for all populations? So the idea is we could take this huge cluster here, 17, train a polygenic score on that, and then we could just test it on every single other cluster and see how well the polygenic scores transfer. So this is some work I did with Shadi Zabad, who is a PhD student with Simone Gravel. So for height, you can see here the y-axis is basically the accuracy of the polygenic score, and then the x-axis is the fixation index, which you can kind of think of as a genetic differentiation. But you can see that there's this downward slope. So for height, as the populations become more genetically differentiated, the less accurate the polygenic score is. But for LDL cholesterol, this wasn't the case. So you can see that it's actually kind of scattered. And it turns out there's a little bit of literature about this where LDL cholesterol is known to be relatively non-polygenic. So there's a few variants that have a very strong effect on the phenotype. But this is a really cool way to look at it because populations are kind of tough to define. And this gives us, in principle, a method to do it for a bunch of analyses. I also use clusters for QC for my data. So I did some work with Georgette Fimmerling, who's I think she may have graduated, but she was a master's student. And we were looking at the Cartesian data set, which is based in Quebec, and I generated a bunch of clusters for Cartesian. Then I started looking at the countries of birth. And this one was really interesting to me because Algeria, Tunisia, and you can sort of, it's a little blurry, but there's France in there too. These all kind of make sense as one cluster because they're geographically near each other. And then there's also the French colonial history. American Samoa is not in any way related to those. So I actually scoured the history, like, was there a French colony in American Samoa? Was there anything like that? But as it turns out, this was a coding error in a questionnaire. And I think it was just an off by one error in the country codes. So I think all of these individuals were also Algerian. But this would have been really tough to find without doing the sort of visual QC. And setting that aside, the purpose of this cluster that we were looking at was for potentially doing downstream sampling. So you can't see the numbers here, but this made up probably like 10, 15% of the potential sample. So that's a huge sample that would have been lost in that case. Um, and one other application is studying environmental variables. So this one here is we take the same approach as we had before with the smoothing of the phenotypes, but rather than using phenotypic data, we use something like smoking behavior, whether or not someone smokes. And we can overlay the smoking behavior on a population level and then study this correlation between genetics and environment because a lot of individuals, they might not smoke themselves as well, but they might be in a population that tends to smoke. This is tough to measure otherwise, and it's also kind of tough to get your mind around. But you can see here there are patterns. So, the darker clusters, these tend to have higher than average smoking rates. The lighter clusters tend to have lower than average smoking rates. So you can do a lot of very interesting stuff, find a lot of really cool correlations between genetics and the environment. So to sort of summarize this section, the UMAP is a huge utility. It's very fast, it's unsupervised, it's flexible, it's label agnostic. You can just run it straight on your data. And biobanks are extremely complex and rich in signal, but I think UMAP is a really great tool for doing these sorts of biobank analyses. And the other thing is it pairs really nicely with an established clustering algorithm, HDB scan. And as you can see from all the examples I've given, it's got a really diverse set of applications. And I know it's been a lot of content to throw at you, but I want to also say that this is like not even close to all the stuff I've looked at. So when I say it's got a diverse set of applications, like it's really got a lot of potential. Um, and I think this is an important point too, that as a scientist, your vision, your intuition, your curiosity, these are all really powerful things to have. So it's seen a lot of rapid adoption in population genetics. Um, and I think because we are so intuitively visual, that it is nice to be able to look at our data. But in terms of the limitations of UMAP, there's still quite a few of these. These are important to mention. It can be tempting to assign meaning to visualizations. There's this great phrase, the territory is not the map. So, if your GPS tells you to drive straight because there's a road in front of you, but your eyes tell you, hey, there's a lake in front of me, I shouldn't drive into that. You should probably take a step back and say, okay, the map is just a visualization of the territory. It's not the territory itself. So, in UMAP, clusters might just be artifacts of data preprocessing. You might have some sort of interesting sampling that you hadn't thought about. So, it's a great first step in the analysis. It should not be the last step. There's no interpretation of long-range distances, and this is because it's not designed to preserve those. UMAP is a neighborhood graph method, so if you tell it to preserve the 15 nearest neighbors, it's going to focus on those. If you have 100,000 points, the 10,000th neighbor is not going to make a difference when you've specified 15 neighbors. You can also have distortions that appear from dropping dimensions. So, if you go from UMAP to 3D, from 3D to 2D, you're going to have some differences in your data. So clusters can tear, they can float around, all sorts of stuff like that. And the parameters matter, especially the number of neighbors and whoops, sorry. Parameters matter, especially the number of neighbors and the number of input PCs. So lastly, I want to talk about the social and ethical implications of some of this work. So how do we define populations? Because in population genetics, we need to define a population. So one way to do it is the minor allele frequency, if that's what you're interested in, or you could be interested in defining a population for epidemiological study. But we do also understand that human genetic variation is continuous, and when you have a discrete category, it flattens this continuity and this diversity, and it oversimplifies it a lot. And the genetics of race and ethnicity, this is a sensitive topic, and rightfully so. If you read the history of science, in particular statistics or genetics in the last 150 years, the shadow of eugenics is very long. Um, one of the things that we want to do is to avoid conflating genetics with concepts like race and ethnicity, which are basically social ideas. So when you talk about how populations are defined, on the left here in the UK Biobank, this is the questionnaire that you have, and it actually just asks you what is your ethnic group, and then you would hit a button that says, white, for example, and then it would have a follow-up question that would say like, what is your ethnic background? So I think the options there are like British, Irish, other, or something like that. So where did these come from? These are basically census labels. So I think this is from like the 2001 UK census or something like that. But these are not genetic labels. These are just like administrative data labels that have been around forever, and they got pulled into the questionnaire and they started getting used. Their use as a proxy for genetics is very strongly discouraged because they're just not accurate. It doesn't make sense. Like if you think about it for a bit, a label, if you were like I was born in Cuba and I grew up in Canada. Um, so genetically, like I would probably be kind of different from a lot of Canadians, but my ethnicity would still be white. So the two concepts are not they're not interchangeable at all. And a lot of this a lot of studies still do something like, oh, in order to control for population structure or to control for homogeneity, we limited the study to the white British population. But you think about it, like you're doing genetic work, but you're basing it off of a questionnaire label. Um, so in terms of how are populations often represented in genetics work, and the idea is usually you just lay the census labels on top of some sort of data that you're working with. So this figure that I have on the left, that's actually what I did in 2019. So sometimes people will use race or ethnicity, or they have this term SIRE that's coming up, which is self-identified race or ethnicity. Um, just to take a step back for a moment, I don't like to use the phrase self-identified because usually it's not like a free form fill in your thing. It's like, here's 5 options. Which one are you? So I use selected. This is a minor thing. But that's typically how it's done. And these variables, they're often correlated with population structure. But this is because, population structure is basically the result of non-random mating. So, it tends to be more related to geography than anything else. And population structure also goes super, super fine scaled. You can find population structure down at like the city level very frequently. So, obviously, city is not very popular as an administrative label because you have like 700 of them, but this is the sort of thing that comes up a lot in these correlations. Um, and these labels, they can be useful to study the biobanks, but it feels reductive. And it's, I think it's not great, but it is, or possibly was, it might be going out of favor now, standard practice when dealing with biobank data. But one thing to emphasize is there's other ways to characterize this data. So, I like to look at the geography. So you can see here, there's huge, obvious, cool patterns in terms of the geographical distribution of people. But this is genetic data that I've colored in by geography. So I think this is really a fascinating way to look at population structure. And instead of using one label to say, oh, like this is the population that we're looking at, I like using the word clouds because it actually breaks these clusters apart and says, by the way, here's an idea of what these clusters represent. Because these are representations of data. They're not the actual sort of phenomena. They're just useful ways for us to understand our data. So this brings us to the All of Us research paper, which, sorry, I'm just going to take a sip of water. So this was published last February. And All of Us is an American data set of about 250,000 people or 300,000, something like that. But their flagship genomics paper was published last year. What they did was they used UMAP, and they generated, this is a subset of the figure, but they generated a UMAP, they split it into 2 UMAPs, and then they colored it by race on the top and then by ethnicity on the bottom. And they got a lot of flack for the figure. Um, which led to a huge debate over UMAP and population genetics because a lot of people were saying like, oh, this is, it's reductive, it conflates race, ethnicity, genetics, and there's been quite a bit written about it at this point, and I think those are valid points for sure. I personally didn't like the figure because I think it didn't show anything super useful. Um, but a lot of people kind of said like, okay, well, you know what? Maybe we shouldn't use UMAP at all in population genetics. Maybe we should just use PCA because PCA is really great. And should we use PCA alone? So, okay, let's say we do PCA on the 1,000 genomes. Oh, we get this little triangle shape. It's really interesting, and it reflects the continental ancestry. Or if you do it on the UK Biobank, okay, we ran PCA, we get this interesting triangle shape, and it represents continental ancestry. Okay, and we'll run PCA on Cartesian. It makes this nice triangle, and it gives us this continental ancestry. So there's kind of 2 questions that come up here. And the first one is people were saying like, oh, we should just use PCA because PCA, it's interpretable, it's harder to misuse and all this. But then the second point is like, are these figures useful? Because, you know, if you gave me a biobank and you told me to run PCA on it for a figure, I would just draw this triangle and then I would just say, here you go, it's done. Like we know exactly what PC1 and 2 are going to look like most of the time. Um, but to address this first point, PCA is also not immune to being misused in visualization. So on the left here, this is I think he's like a white supremacist on Twitter or something. But basically, he took a PCA figure and then kind of made this weird argument that encouraged inbreeding, so that way white people would be closer to each other in PCA, which is like a bonkers thing to suggest because it's like there's like 80 things wrong with it. Um, but a lot of people argued for using PCA over UMAP because it's this idea that it's more interpretable, it's linear, it's deterministic, all these other nice properties. But obviously, it can still be misused. And in fact, these PCA plots are very frequently misused by racists. And they've actually appeared in manifestos justifying violence, racial violence. So this was work by Jedediah Carlson on countering the weaponization of genetics research by extremists. And my current supervisor, Sohini Ramachandran, she's a co-author on that. But we've worked a lot to think about like what is going on here in terms of genetics research being used by extremists. But I think about, okay, well, what is the purpose of a figure in the first place? It should communicate something useful. So some of the work I've been doing with all of us is, okay, instead of coloring it in by, say, census labels, why not color it in by zip code? So we could look at population structure versus geography. So you can see here, like there's patterns related to zip code. And this is some work I've been doing with Siobhan Holness, a PhD student. Um, but I think there's a much broader issue that's being ignored, and this is this idea of genetic essentialism in race science, and it's this pseudoscientific belief that racial and ethnic groups are genetically distinct. This is not supported by genetic research. In fact, we find the opposite very, very often because it just makes sense if you think about it. Um, but is that the message people are taking away from genetics research? So there's a lot of work by Aaron Panofsky, who's written quite a bit about this. But my question is the full body of genetics research, is it fueling genetic essentialism? We might say we don't want to fuel that, but how would we study that? And I thought, okay, well, maybe we could use Wikipedia as a data source. Because one day I was looking at the article on Cubans, and then I noticed, oh, there's a section here about the genetics of Cubans. And I realized that this section's like autosomal or distributions of different variants, like admixture plots, things like that. But lots of pages about populations now discuss genetics. So pages about nationalities, ethnicities, religious groups. And I wonder, like, how common is this? What are they saying? What sort of research is being cited? So I've been doing some work with a PhD student, Abby Kunzelman, and the idea is we took a whole bunch of Wikipedia pages and used the Wikipedia API to collect data about them going all the way back to 2001. So we focused on pages about populations, so demonyms, which is a fancy way of saying like a name for someone from a place. So, you know, Canadian or the demonym from Canada is Canadian. For France, it's French people. For Korea, it's Koreans. It's like a nationality, but it's more general. And we looked at all the pages that were also categorized on Wikipedia as ethnic groups. And then we performed text mining on strings to look for genetics related words. So things like haplo, admix, genome. And we collected about 2 million revisions from 5,000 Wikipedia pages. So I can skip through this in a moment. But yeah, one of the takeaways was that genetics is now very, very common in terms of the discussion of populations. So if you're reading an article about populations on Wikipedia, about 20% of these pages now have terms related to genetics in them. So when we're talking about, human groups, genetics has become kind of the common lexicon. And in fact, about 45% of pages about demonyms, so like nationalities effectively, now reference genetics. So these appear in different contexts. So sometimes you'll see a phrase like ancestry or origins or ethnogenesis, which is like, how did this ethnicity come to be? And this is just a graph showing that over time, the genetics terms have really risen linearly over the last 20 something years. But yeah, so like an example is the Wikipedia page on Italians. You can see they have a section on ethnogenesis, but they have a principal components plot. They have all of this research about like ancient DNA, about admixture, about all this other stuff. The Wikipedia page on Spaniards, there's a section on historical origins and genetics. So whether we like it or not, like genetics research is being used to possibly even define populations, even though we don't want people to do that. And in fact, when you look at nationalities, um, I think like almost, yeah, so almost half of these, they now have genetics discussions. So Canada, for example, the page on Canadians does not discuss genetics at all, but the page on Americans does. French people, Spaniards, Russians, Chinese people, Australians, all of these do mention genetics in some way or another. So this is an important thing to study. So this is, I'm at the start of this project, but I think this is a really important thing to look at because we talked about, because, last year there was this controversy about, oh, how do we visualize genetic data? And I think it's important to take a step back and say, like, how's genetic data being used in general? All right, wrapping up here, future work and conclusions. For the dimensionality reduction and clustering, right now I'm doing some interesting stuff on studying gene by environment interaction, interpreting the clusters in terms of demographic processes. So, did this cluster arise because of migrations or admixture or something like that? And looking at using UMAP and clusters for sampling and epidemiology. And the social and ethical implications, I'm really interested in doing a more in-depth analysis of some of this Wikipedia work that I've been doing. I'd like to do a temporal analysis of the text and also identify potentially influential research papers, biobanks, things like that. So yeah, in conclusion, UMAP and HDB scan, really powerful tools for biobank analysis. There's many approaches beyond like just making a figure. It's really efficient for interrogating your data, lots of potential to study intricate relationships. And also, genetics is becoming more common when discussing group identity, and I think this is something really important to keep in the back of your mind working with genetic data. So with that, whole lot of people to thank, and also the QLS program. Miss all you guys. Um, and yeah, I can wrap up there. So if there's any questions, you can email me at either of these. And if you're interested in my papers, my code, my thesis work, you can visit my GitHub at this link. So thanks a lot.
Apologies. Sorry, I was muted. Questions from the room or on the chat? You can put them in the chat online. Bangley, please.
Actually, the part of
Bangley, can you speak up so they can hear you?
actually the part of social media, like you mentioned, Wikipedia something. I think there's one part I missed. What is the goal of that part? You check the terms online on Wikipedia or something. What's the goal?
So the goal is to study the role of genetics research in the information ecosystem. So there's been a lot of work being done on I guess they call it the weaponization of genetics research. And one of the things that happens is all of our research is online. It's often open access for free. It's very easy to access. And the good thing about this is it means great. Everyone has access to scientific research. Um, the downside is, the internet's a big place and there's a lot of bad actors on there. There's a lot of people who want to, for example, extract snippets of text or things like that to sort of push a narrative that supports genetic essentialism. But it's hard to measure this kind of stuff. So, one of my motivations on the Wikipedia work was to say, okay, taking a step back, like, how is our research being used in general? Like, we don't know how the public really understands genetic research because I don't know if you've ever done this or if you've traveled to a conference and, you tell the taxi driver, like, oh, I'm on my way to a genetics conference. I have to hold my breath because I don't know what the next question that they ask me is going to be. And it's not like, um, I've been working on this stuff for 6, 7 years in a very technical manner. I'm interested in studying as in addition to the actual like genetic stuff, how are people interpreting this research? And since Wikipedia is one of these huge resources, it's used for large language models. It's the one of the top 10 websites, and it's generally where people go to have a question answered. Um, if somebody asks a question like, genetically, how many Arabs or types of Arabs or something are there? And this is stuff I've asked to, I think, Microsoft Copilot or an older version of ChatGPT, and it would say something like, there are genetically 4 types of Arabs based on genetics research. And then I would go back farther and farther, and I would trace it back and find like, oh, okay, it's because, there was a genetics paper from like 2011 that said we ran admixture and we found that it was optimized at k=4, which then gets put into a Wikipedia article where someone added it in there saying like, so and so et al. In 2011 found 4 clusters of Arabs in North Africa or something like that, which then gets pushed all the way downstream. So the goal of this is for me to just kind of measure, first of all, how often is genetics being used in sort of this public-facing repository of knowledge. But on top of that, what's the context? Is it being used to define groups? So for example, like the average French person has this much admixture or something like that. Or is it being used more to support, like, genetic research shows that there were migrations in like 1700 or something like that. So that's kind of the broad motivation for it.
Okay, but there's one problem in China. I don't think people can get access to Wikipedia directly. How do you get the data from China?
So I can't get the data from China, unfortunately, and this is true. Wikipedia has been banned in mainland China since like, I don't know, 2016 or something like that. I mean, at the moment, this is just kind of limited to what I have, which is the English language Wikipedia. There's a bunch of biases built into that. It's a very, obviously it's English. It's very heavily skewed towards American points of view. It also tends to be very skewed to sort of the Bay Area kind of worker because that's where it was started. So it's like 90% men that write it. So there's all those biases inherent. So I can't speak to how, for example, a mainland Chinese individual would interpret this stuff. But in terms of like an American-based English language source, I think that's better.
Yeah, actually in China, those things are in Chinese, and the database is Baidu Baike. It's another searching engine.
Hmm.
But I think that one may be too hard for you to get the thing because it's all in Chinese.
Yeah, and my Chinese is pretty non-existent. I don't know how their API is, but perhaps at some point in the future we could talk.
Other questions? Fred?
Yes, Alex, that was a really nice talk. So you're selling UMAP as working great for biobank. So does that mean that it doesn't work as well for other type of data? I'm just curious about the limitations of UMAP if you came across areas, types of data that it doesn't perform so well.
Honestly, I haven't personally used UMAP on a ton of other data, but I know quite a few people that have. Like single cell is the most obvious one that comes up. Single cell people use UMAP all the time, which has its own debates, but it's still fantastically useful for that. But I've seen UMAP used on pretty much everything. I've seen it used on like NBA basketball players to kind of cluster them by performance. I've seen it used on actually on Wikipedia text itself to kind of find clusters in there. I know it was actually originally developed for sort of national security purposes because it was used to cluster text from emails to find phishing emails, and it worked pretty well for that. So, like the key limitation is, I think generally data set size is one of the things. So if you have a data set that's under like 300 or 400 points, it might not work so well. Um, but I haven't seen anything where it just doesn't work at all. But I also haven't really applied it to a whole ton of data sets.
Thanks.
I don't see any other questions online. I don't see any other question. Oh, yeah, from Suresh.
Oh, yeah.
Can you see that, Alex?
Yes. So the question is, emphasizing that this is a naive question. I understand the we don't want weaponization part very well, but what is your take on the genetic correlations that are present, sometimes strong, and clearly identify subpopulations, geographic or linked by selective marriage, since these may be relevant for population health, for example? Those are worth studying for sure. I mean, it's kind of depends on what is the so depends on the purpose of the analysis, either in terms of what you publish or what questions you're asking. Um, so, like, if you find genetic structure, it's relevant for sure, but it depends on what it is that you're saying about it. So, I know I've seen someone who used UMAP, and they showed me at a conference that they were working on the Indian subcontinent, and they found that UMAP actually separated a lot of populations based on language and caste really well, which, that has um, the analysis that you want to do on that for something like population health is, from my perspective, often the first thing I would do is like, okay, setting aside the population labels that we have, what if we overlaid something like um, air quality, water quality, where they live, access to education in the United States, something like access to health insurance. I think that's actually a lot more informative a lot of the time. So, it's I'm not going to discourage people from, yes, cases are intermixed geographically very strongly in India. Yeah. So, a lot of the structure population structure is at its core a phenomenon of non-random mating, and generally the way that people choose their mates is they are geographically nearby. Like it's pretty rare for people to just like meet from a long distance and then stay at a long distance. So I think the thing to keep in mind is that geography is actually kind of the biggest driver of a lot of it. And that geography correlates with so many things. And there's a huge focus on variables like race and ethnicity because they're so prevalent in society. But I think the more informative ones a lot of the times are actually a lot of the social factors or environmental factors. So it's, you know, there's a responsible way to sort of approach these kinds of analyses. And if you just come out and say like, okay, we did these like 5 clusters and the 5 clusters just have, I don't know, like different educational attainments and you just leave it at that, I don't think that's a very useful analysis. But if you go in deeper and you say, oh, it turns out that there's differences between environmental factors and these environmental factors we know are related to something like asthma, I think that's a good way to approach it.
Yeah, thanks, Alex. It's a long discussion, but India may be a special case. So
Yes.
labels don't, because of very, very strong social pressure for selective inbreeding within castes. So the labels will not match as well as the caste labels.
Yeah.
They all share the same environment, the same air, the same
Yeah.
border. So yeah, but I understand what you're saying. Thank you.
Yeah. All right. Thanks.
Okay, well, thank you, Alex. That was a wonderful presentation.
All right. Thanks for having me. All right. Always a pleasure. And everybody enjoy your Tuesday. And yeah, if you